So yesterday we have seen little bit about the sterilization. Yesterday we have started with the sterilization. Sterilization is nothing but the absence of all forms of organisms. So whether vegetative or spores. So the process of removal or killing of all viable forms of organism is known as sterilization. Sterilization क्या होता है? Removal of all microorganism, removal of all different types of or all forms of microorganism, whether it is a live, whether it is a non-live, whether it is a spore. So removal or killing of all forms of organism from your preparation known as the sterilization. So removal, killing of all viable forms of microorganism including their vegetative form and spores known as a sterilization. So what is aseptic technique? Aseptic technique is nothing but it is a technique by which you use to prevent the contamination in your product. So aseptic technique is the process, or it is a in which all possible contamination of microorganism can be prevented to get a to get entered into your product being processed. तो आप जो प्रोडक्ट बना रहे हो उसमें पहले से ही माइक्रो ऑर्गेनिज्म का कंटेमिनेशन आने ही मत दो. ऐसी टेक्निक फॉलो करो कि पहले से ही आपने एयर फिल्टर्स स्टेराइल एयर प्रोड्यूस करो, क्लास 100 एरिया प्रोड्यूस करो. So you can process parental products terminally sterilized but not terminally sterilized. So those products which are made by aseptic technique, so they are processed in the aseptic environment only. That is nothing but the technique in which all possible contamination of microorganism can be prevented to get enter into your product. So we have already seen this, but I am repeating it. Disinfection. So what do you mean by disinfection? It is nothing but the process of destruction or killing of organisms, pathogenic organisms that are capable of giving rise to the infection, that are giving rise to infection. तो disinfectant क्या होता है? Method, it is a destruction or killing of microorganism which gives rise to the infection. But disinfectant it is applied to a non-animate objects. You can say inanimate objects. Disinfectant could apply करता पण disinfectant they are applied on a floor, they are applied on a utensil, they are applied on a table, etc. Disinfection is a process of killing of microorganisms again capable of giving rise to the infection. So chemical disinfectants are used. So now there are two terms disinfection. The agent which are used to for the disinfection known as a disinfectant. Another term is there known as a antiseptic. So disinfectant means these are the chemicals which are applied on inanimate object for killing of microorganism. Antiseptics are the chemicals or agents which are applied to a skin and mucous membrane to prevent or kill the growth of microorganism. So you must know the definition, you must know the difference. Method of sterilization, there are different methods. Yesterday we have seen. So first one is a physical method. So you can also sterilize by using sunlight or by using drying. So your grandmother, your parents used to sterilize the grains stored in the house in the summer using a sunlight. उन्हाळ्यामध्ये तुम्ही ज्वारी, बाजरी, गहू जे असेल ते गच्चीवर उन्हात ठेवता का for the sterilization, because that sunlight has a UV rays and that will kill the microorganisms. Right? Now dry heat. So dry heat is used in the flaming incineration. What here yesterday I have told you. Moist heat is also there because steam has a more penetration power than dry heat. So they cause the oxidative damage to the bacteria. Moist heat causes coagulation and oxidative damage to the bacteria. So moist heat you are using in the pasteurization, boiling steam under reduced pressure or steam under normal pressure, right? Then filtration is there. So filtration is a mechanical method of sterilization. So also known as a cold sterilization method. Filtration. So filter candle, asbestos pad, membrane filters. Membrane filters are made from a cellulose. Remember, asbestos are nothing but carcinogenic. Radiation sterilization, in the different radiations are used. Ionizing radiations and non-ionizing radiation. UV, IR, they are known as a non-ionizing radiation. But alpha rays, beta rays, gamma rays, they are known as ionizing radiations. So ultrasonic and sonic vibrations are also used for sterilization. Next is the chemical method of sterilization by using chemicals. Different chemicals like alcohol, aldehydes. So they are used alcohol, aldehydes, phenols. So alcohol like ethyl alcohol, isopropyl alcohol, that is the most commonly used in our industry. Trichlorobutanol, then aldehyde in that formaldehyde, butyraldehyde, they are used. Surface active agent like surfactant, they are also used as a chemical disinfectant, right? So gases like ethylene oxide, formaldehyde, beta propyl lactam. So yesterday we have seen this in detail. That's why I am revising it very fastly. I hope I am audible to you clearly. Sterility is nothing but the absence of any viable form of organism. Sterility means what? It is the term. It is nothing but the total absence of any form of microorganism. Antiseptic is nothing but the chemical which is applied into a skin or mucous membrane or simply in a human body to prevent the or kill the microorganism. Disinfectant, it is nothing but they used to kill the microorganism and it is applied to an inanimate object like floor, tables, etc. Now there are few terms which are used to indicate the sterility कि माइक्रो ऑर्गेनिज्म मरे या नहीं है उसके लिए कुछ टर्म्स है उसके ऊपर बहुत बार क्वेश्चन आ चुके हैं. So first term is nothing but the decimal reduction time. Decimal मतलब क्या है? Decimal मतलब 10. Decimal का मतलब ये होता है 10. Decimal reduction time or it is known as a D value. So it is a time of disinfectant required to reduce the population of microorganism by 90%. कि कोई फिनॉल है कोई अल्कोहल है उसको कितना टाइम लगता है एक 100% कॉलोनी है बैक्टीरिया का उसको 90% किल करने के लिए कितना टाइम लगता है दैट इज नोन एज द डेसिमल रिडक्शन टाइम और डी वैल्यू. It is nothing but the time of disinfectant required to reduce the population of microorganisms by 90% or simply one decimal, that is 90% of one logarithmic units. So decimal reduction time question इसके ऊपर आता है what is D value, what is what is D value, what is then Z value, what is E value. So next term is a Z value. Z value is nothing but the thermal destruction time. Thermal destruction time. So Z value thermal destruction time. So thermal destruction time of disinfectant, that is nothing but the degree of temperature required to reduce the 1 log of D value or simply 10-fold changes in D value. इसके ऊपर क्वेश्चन आ चुका है कल भी बोला था मैंने. So thermal destruction time is nothing but the Z value or it is the temperature required to reduce the 1 log of D value or 10-fold changes in the D value, right? So decimal reduction time, thermal destruction time. Next is the methods of sterilization. Different method of sterilization we have seen. So dry heat sterilization. Dry heat sterilization is done by using hot air oven. And what is the mechanism of dry heat sterilization? Question आता है उसके ऊपर से. So dry heat causes the oxidative damage and denaturation of bacterial protein. Bacteria मरता कैसे dry heat से? So there is oxidative damage to the bacterial enzymes and denaturation of bacterial protein. Now what is the holding temperature? Again important. So holding temperature for dry heat sterilization. So 180 degrees Celsius for 2 hours or 260 degrees Celsius for 45 minutes. So what is use of this dry heat? So dry heat is used to sterilize the glasswares and other metal instruments or simply powders, oily injections. So they are dried by dry heat sterilization. तो उसका एप्लीकेशन के ऊपर भी क्वेश्चन आता है. Application पण महत्वाचा आहे. So application is also, so remember the application. Then next one is a moist heat. Next one is a moist heat. So as I told you, moist heat means you are using steam. Now that steam has a more penetration power than your dry heat. That's why moist heat sterilization is more effective than dry heat. Now in that you are using a apparatus known as a autoclave. Now what is the mechanism of action of dry heat? What is the mechanism by which bacteria or pathogenic organisms they are destroyed by moist heat? So obviously there is a coagulation of bacterial protein, denaturation and coagulation. Main mechanism is a coagulation of bacterial protein. Denaturation is also there, but main mechanism for dry heat nothing but the oxidative damage and denaturation. तो moist heat में क्या है? coagulation of protein. देखो आप अंडा खाते हो, अंडा उबालते हो अंडा इनिशियली लिक्विड होता है, अगर उसको ब्रेक करके देखो तो लिक्विड है उसमें, बट जब आप बॉईल करते हो उसको बाय यूजिंग स्टीम, दैट लीड्स टू कोएगुलेशन ऑफ प्रोटीन. कोएगुलेशन और डिस्ट्रक्शन ऑफ प्रोटीन मीन्स क्या कि लॉस ऑफ नेटिव स्ट्रक्चर ऑफ प्रोटीन, दैट इज नोन एज अ डीनेचुरेशन ऑफ प्रोटीन. अपन ने देखा है प्रोटीन चैप्टर में, loss of native structure of protein is known as a denaturation of protein, and coagulation is also one of the type of denaturation. So main mechanism for moist is nothing but the coagulation, but right, it is nothing but the oxidative damage and denaturation of bacteria. So what is the holding temperature in the autoclave? So there are different holding temperature, but remember 115 to 117, 115 से 117 डिग्री for 30 minutes. Again 121 डिग्री सेल्सियस for 20 minutes. अपन नॉर्मली कितना यूज करते हैं मॉइस्ट स्टरलाइजेशन में? 121 डिग्री सेल्सियस, 121 डिग्री सेल्सियस for 20 minutes at a 15 lb atmospheric pressure. Okay? So moist heat sterilization is used for the sterilization of IV fluids, rubber and plastic container. Then tyndallization process. Tyndallization. So tyndallization means fractional sterilization. इसको fractional sterilization बोलते हैं. So heating of a that medium under sterilization at 80 degrees Celsius for 1 hour or 100 degrees Celsius for 20 minutes for successive 3 days. 3 दिनों तक आपको पहले दिन 80 डिग्री सेल्सियस के ऊपर 1 आवर के लिए हीट करना है, छोड़ देना है, दूसरे दिन भी करना है, छोड़ देना है, तीसरे दिन भी करना है, छोड़ देना है. That is known as a tyndallization, known as a fractional sterilization. It is used for the surgical dressing and culture media. Surgical dressing and culture media, they are sterilized by tyndallization technique. Remember, ठीक है. Next होता है आपका pasteurization. So Louis Pasteur, this method was invented by Pasteur for the sterilization of milk. That's why. So what is the holding temperature over here? So you have to heat the milk at a 62.8 degree or you can say 63 degree for 30 minutes, known as the holders method. अगर ये टेम्परेचर यूज करेंगे तो इसको अपन बोलते हैं होल्डर मेथड. नेक्स्ट वन इज नथिंग बट द हीट द मिल्क एट 71 to 72 degree for 15 seconds, known as a splash method. Then third one is the heat the milk 140 degree less than 1 second. So it is used for the sterilization of milk and popularly it is called as a pasteurization of milk. थर्ड होता है आपका रेडिएशन स्टरलाइजेशन. तो रेडिएशन स्टरलाइजेशन, तो इसमें भी 3 टाइप के रेडिएशन यूज करते हैं, आयोनाइजिंग रेडिएशन एंड नॉन आयोनाइजिंग रेडिएशन. सो यूवी आईआर जो है नॉन आयोनाइजिंग रेडिएशन. सो यूवी इज वन ऑफ द यूवी. सो मैकेनिज्म भी दिया है उसका. मर्करी वेपर लैम्प इज यूज्ड टू प्रोड्यूस द यूवी रेज एट 253 नैनोमीटर. यूवी का रेंज होता है 200 से 400, 200 to 400 नैनोमीटर. नियर यूवी होता है लेस देन, सॉरी वैक्यूम यूवी होता है लेस देन 190. विजिबल होता है 400 to 800. आपको रेंजेस भी मालूम होना चाहिए. तो व्हाट इज द मैकेनिज्म बाय व्हिच यूवी रेज विल डिस्ट्रॉय द माइक्रोऑर्गेनिज्म? यूवी रेज विल डैमेज द न्यूक्लियोप्रोटीन ऑफ द माइक्रोऑर्गेनिज्म. जो बैक्टीरिया होता है उसका न्यूक्लियोप्रोटीन्स डैमेज करता है. सो इट इज यूज्ड फॉर थर्मोलाबाइल सब्सटेंसेस. जो टेम्परेचर की वजह से प्रॉब्लम होता है, थर्मोलाबाइल heat sensitive substances. It is used for maintaining aseptic technique, also used for the hospitals, and UV chambers are when UV chambers are available in the hospital. Then ionizing radiations, ये ions बनाते हैं, beta rays, gamma rays. So denaturation and damage to the DNA and enzyme. So what is the mechanism? So denaturation and damage to the DNA, enzymes and other component of bacterial. So formation of free radical. So they will form a free radicals like gamma rays and beta rays. They are used for the sterilization. So they are again used for the thermolabile substances, sterilization of syringes, then plastic catheters, needles, surgical dressings and blades. Third होता है आपका gaseous sterilization. So here you are using gas. So gas chemically react with the bacterial cell. So ethylene oxide is used, formaldehyde is used, then beta propanolactone. These 3 gases are used. All they are known as alkylating agent. They will cause methylation, okay, methylation or propylation of the sulfhydryl groups of protein, bacterial protein. तो उसको alkylation करेंगे तो alkylating agent है. They will destroy the protein, right? because after alkylation the bacteria will not utilize that protein. So again it is used for the thermolabile substances, plastic and rubber, fumigation of operation room, sterilization and fumigation of aseptic room. Fumigation के लिए मैंने बोला था, so most of the hospital में क्या होता है कि before surgery वो क्या करते हैं, फॉर्मेलिन का सोल्यूशन होता है, पोटेशियम परमैंगनेट होता है, वो दोनों को मिक्स करके रूम में छोड़ देंगे और बाहर निकलेगी 1 hour वैसे ही रख देंगे. So formalin react with the potassium permanganate to release of formaldehyde fumes, and that fumes will sterilize whole operation theatre, right? So that is the gaseous sterilization. Am I audible? Am I audible to you? Or sterilization by filtration. So sterilization by filtration, इसमें क्या आता है? Sterilization by filtration, इसमें we are using bacteria proof filters. We are using bacteria proof filters, means these filters they will remove the bacteria. So bacteria proof filters. So sterilization by filtration, also known as a cold sterilization, also known as a cold sterilization. So in that filter candles are there, then asbestos filters are there. So filter candles, unglazed ceramic filters, diatomaceous earth filters are there. Asbestos filters क्या होता है? They are carcinogenic in nature. Sintered glass filters होता है, तो prepared by using fusing, prepared by using or prepared by fusing powdered glass particle, finely powdered glass particle जो होता है. So you have to powder the glass particle, fine powder of glass particle they have to fuse, and from that sintered glass filters are made. Membrane filters they are made from cellulose, okay, and other polymer. Then radiation sterilization होता है, non ionizing radiation. So low energy type, लो एनर्जी टाइप में यूवी आईआर होता है, दे डैमेज द जेनेटिक मटेरियल ऑफ बैक्टीरिया. आयोनाइजिंग रेडिएशन जो होता है उसमें एक्सरे, गामा रेज एंड कॉस्मिक रेज होता है. दे आर हैविंग हाई एनर्जी, सो दे हैव अ लिटिल इफेक्ट ऑन द जेनेटिक मटेरियल एंड अदर वाइटल component of microorganism, but they will kill the microorganism. They are known as a sterilization by filtration or cold sterilization. Next, chemical sterilization. So we are using alcohol. Okay, so alcohol is used as a disinfectant. So for in corona crisis also there was a lot of sanitizer known as alcohol based sanitizer. So alcohol in that ethanol and isopropyl alcohol they are most frequently used. तो अल्कोहल कैसे मारता है बैक्टीरिया को? So alcohol they act as a denaturation of bacterial protein. They act by denaturating the bacterial protein. They have no action on spores. ये spores को नहीं मारेगा. Alcohol will only kill the live bacteria. Next होता है formaldehydes. So aldehyde में formaldehyde is most commonly used. So which will interact with the amino acids and proteins of the microorganism. Mostly used to preserve the anatomical specimens. अगर आप कोई आपको देखा होगा 12th में भी ब्रेन वगैरह प्रिजर्व करके रखे आपके कॉलेज में. So for that preservation formaldehyde or formalin solution is used. Formaldehyde gas is widely used for the disinfection of operation theatre. तो फिनॉल होता है, फिनॉल आप फिनॉल यूज करते हो घर में भी to clean your pores. So phenol exerts lethal effect on the microorganism because of their capacity to cause the membrane damage. जो bacterial cell membrane उसको damage करता है and release the cell content. Ethylene oxide. So mechanism it causes the alkylation of amino carboxy or hydroxy or sulfhydryl groups present in the protein molecule of bacteria. जो halogen होता है chlorine, bromine, iodine. So iodine is used as a antiseptic. Iodine is a strong oxidizing agent and it is used as an antiseptic on skin and mucous membrane. Chlorine is nothing but the strong oxidizing agent again, and it is used for the disinfection of your water. अगर municipal corporation का water पीते हो उसमें chlorine डालते हैं. It is oxidizing agent. It will kill the bacteria present in the. Then heavy metals. So heavy metals will also cause denaturation of enzymes and other essential component of protein. Surface active agent जो surfactant होते हैं anionic, cationic and non-ionic. So anionic, cationic detergents causes the disruption of enzymes, disruption of enzymes, bacterial enzymes. Then protein denaturation and disruption of plasma membrane. So आपने देखा होगा सिंपली कोरोना के पीरियड में बोला था अगर आपके पास सैनिटाइजर नहीं है सिंपली सोप वाटर यूज करो. Why soap water? because soap water consists of a surface active agent and that will also kill the bacteria by this mechanism, protein denaturation or plus disruption of plasma membrane or disruption of enzymes, right? So methods for sterilization and their validation okay, respective validation, chemical, biological and chemical, biological and physical later. So आपने sterilization किया, but आपको क्या मालूम whether sterilization is complete or not? So you cannot observe by naked eyes because bacteria they are not seen by naked eyes. So for that you should have some indicator. जैसे आप analysis करते हो तो आपको क्या मालूम कि reaction complete हो चुका है या नहीं, आप indicator use करते हो उसमें. जैसे इंडिकेटर आपका चेंज कलर चेंज दिखाएगा, मतलब क्या कि रिएक्शन कंप्लीट हो चुका है. तो वैसे ही स्टरलाइजेशन के लिए इंडिकेटर होते हैं. सो वेरी वेरी इंपॉर्टेंट क्वेश्चन बहुत बार आ चुका है इसके ऊपर, तो ये स्टडी मैंने बोला होगा आपको, बट स्टिल. सो फर्स्ट मेथड ऑफ स्टरलाइजेशन इज मेथड ऑफ द ड्राई हीट स्टरलाइजेशन. ड्राई हीट स्टरलाइजेशन. तो यहाँ पे इंडिकेटर जो होते हैं तीन टाइप के होते हैं, फिजिकल इंडिकेटर, केमिकल इंडिकेटर एंड बायोलॉजिकल इंडिकेटर. Physical indicator. So recording of temperature by physical means. That is a physical indicator. Temperature charts. So it is a physical indicator for dry distillation. Then chemical में आता है Brownies tube. Brownies tubes. Question आ चुका है इसके ऊपर. Brownies Brownidic heat sensitive tapes. They are used for the chemical indicators. Validation of dry distillation. Biologically, spores of Bacillus subtilis. Question आ चुका है इसके ऊपर. Bacillus subtilis के spores they are used for the validation of dry distillation. Then spores of Clostridium tetani they are also used. Question बहुत बड़ा चुका. Now moist heat sterilization में physical, chemical, biological indicator कौन से? So biological indicator for moist heat sterilization again spores of Bacillus stereothermophilus. Question आ चुका है. Clostridium sporogenes. Clostridium sporogenes. So they are the biological indicator. Okay. Then autoclave tapes are there. Autoclave tapes, thermocouples. Autoclave tapes they are the chemical indicator, and thermocouples they are the physical indicator. Then sterilization by radiation उसके लिए कौन सा indicator है? Chemical dosing metals are used as a chemical indicator, and biologically Bacillus pulmis and Cephrecus they are used as a biological indicator. Bacillus pulmis and Bacillus cephrecus they are the organism which are used as a biological indicator. तो ये भी क्या है? spores of Bacillus pulmis or spores of Bacillus cephrecus. Okay. Then gaseous sterilization. So here Roche's sachet indicators are used, which are chemical indicators for gaseous sterilization. For biologically, spores of Bacillus subtilis they are used as a biological indicator. ये ऑलरेडी हो चुका है इसलिए फास्ट ले रहा. स्टरलाइजेशन बाय फिल्ट्रेशन. Pseudomonas dimetura serratia mersens. Pseudomonas diminuta. Pseudomonas diminuta and Serratia mersens. They are the spores of these bacteria. They are used for the sterilization by filtration as a biological indicator. So again, physically membrane filter integrity they are checked by bubble point test and forward flow test. These are the indicators. Now again you can see over a sterilization technique. So moist sterilization आता है. Bacillus stereothermophilus and Clostridium perfringens they are the biological indicator. Chemical indicator होता है strip paper. Dry heat के लिए Bacillus subtilis का spores यूज किया जाता है, and brownish tube Brownidic heat sensitive they are the chemical indicators. For gaseous and ethylene oxides, so Bacillus pulmis spores are used as a biological indicator, and Roche sachet are a chemical indicator. Radiation and filtration sterilization में Pseudomonas diminuta, Serratia mersens and Saccharomyces cerevisiae. Question आ चुके हैं इसके ऊपर. They are the biological indicator, and with dosimeters they are a chemical indicator. Then types of brownies tube. Brownies tube के types होते हैं. Type 1 is used for the moist heat sterilization. Only 126 temperature होता है. So color indicator क्या होता है? So when you will get a black spot on this brownies tube, so you can say that the sterilization is completed. टाइप 2 होता है हाई वैक्यूम मॉइस्ट यूज किया उसमें, सो इट इज अ टेम्परेचर 130 प्लस होता है, यू विल गेट अ येलो स्पॉट आफ्टर स्टरलाइजेशन इज कंप्लीट। 3 ब्राउन इज टू होता है यूज फॉर ड्राई हीट, 160 डिग्री सेल्सियस टेम्परेचर होता है, यू विल गेट अ ग्रीन स्पॉट आफ्टर कंप्लीशन ऑफ स्टरलाइजेशन। टाइप 4 जो होता है ड्राई हीट इन एन इंफ्रारेड कन्वेयर ओवन, सो 180 डिग्री सेल्सियस टेम्परेचर पर होता है ये, इसका यूज किया जाता है, यू विल गेट अ ब्लू स्पॉट आफ्टर स्टरलाइजेशन इज कंप्लीट, तो ये इंपॉर्टेंट है। टाइप 1 जो ब्राउनिश ट्यूब है मॉइस्ट हीट में यूज किया जाता है, टाइप 2 हाई वैक्यूम मॉइस्ट हीट में, टाइप 3 ड्राई हीट में, टाइप 4 अगेन ड्राई हीट में, तो ब्लैक येलो ग्रीन ब्लू spot respectively that will indicate the completion of sterilization. Terms used for expression of resistance of bacteria for chemical disinfectant. क्वेश्चन इसके ऊपर लास्ट ईयर भी आ चुका है। Terms used for the expression of resistance of bacteria for chemical disinfectant. पहला टर्म है D value. What is D value? D value is nothing but the decimal reduction. Decimal reduction time. It is nothing but the time required for time required by a disinfectant to reduce a 90% of viable form of microorganism. D value or decimal reduction time. So time in minute at any temperature to destroy 90% of viable microorganism. That is a D value. Example D115 degree Celsius. D value at 1115 temperature. D value at 121 degree. So time required to destroy the 90% of microorganism at a particular temperature. That is a decimal reaction time or D value. Z value क्या होता है? Z value is nothing but the degree of temperature required to produce 10-fold change in the D value. As it is question आ चुका है. What is Z value? It is nothing but the degree of temperature required to produce a 10-fold change in the D value. Z value क्या है? As it is question आ चुका है लाइन के ऊपर. So Z value is the degree of temperature required to produce 10-fold change in the D value. It is nothing but the heat resistance of microorganism to change in the temperature. Bacterial spore will have most of the Z value in the range of 10 to 15 degree. Next to the EF value. What is the EF value? It is a unit of lethality. It has been devised for food industry called as the EF value, and it is nothing but the equivalent in the minute. It is nothing but the equivalent in the minute of 121 degrees Celsius for all, and it is considered with respect to its capacity to destroy the spores as well as destructive cells of particular organism. तो ये वैल्यू क्या है? यूनिट ऑफ लीथैलिटी. यूनिट ऑफ लीथैलिटी ये फूड इंडस्ट्री में ये इसका यूज किया है. This device in the food industry called as the EF value, and it is nothing but the equivalent in the minute of 121 degrees Celsius for all. It is considered as a with respect to its capacity to destroy the spores and waste cell of particular organism. EF value. So D value, Z value, F value. Next is Q10 value. Again, values in Q10 value or temperature coefficient, known as the temperature coefficient or Q10 value. So increase in the killing rate brought about by increasing the temperature of 10 degree Celsius. अगर टेम्परेचर 10 डिग्री से बढ़ा दिया, किलिंग रेट कितना है माइक्रोऑर्गेनिज्म का? इंक्रीज इन द किलिंग रेट ब्रॉट अबाउट बाय इंक्रीज इन द 10 डिग्री टेम्परेचर, दैट इज नथिंग बट द Q10 value. And it is for Q for moist it is around 14, for dry it is 4. Dry it is 4. So मतलब क्या है कि अगर 10 डिग्री टेम्परेचर बढ़ा दिया आपने ड्यूरिंग मॉइस्ट इंस्टॉलेशन, तो 14 टाइम्स बढ़ता है. इफ यू इंक्रीज द 10 डिग्री सेल्सियस फॉर ड्राई रेट 4 टाइम्स बढ़ता है. सो Q10 टेम्परेचर और it is a Q10 value or temperature coefficient known as a increase in the killing rate of the bacteria brought about by increasing the 10 degree temperature. That is known as a Q10 value. Very very important है, ये चीजें आपको मालूम होना चाहिए. Next is the inactive factor. Inactivation factor is nothing but the degree to which viable population of microorganism is reduced by treatment applied it, and often by dividing the initial viable value divided by final viable value. The inactivation factor is the degree to which viable population is reduced by treatment applied. मतलब अगर आपने क्या किया, डिसइंफेक्टेंट का यूज किया, तो जो ओरिजिनल पॉपुलेशन था, वाइबल पॉपुलेशन, उसमें कितना कम हो चुका है. Viable population of microorganism that is being reduced from the treatment, whatever it may be either disinfectant, it may be either autoclaving, whatever, and that is obtained by dividing the initial viable count divided by final viable count. It is known as inactive factor. ये definitions जो है ना important है question है. Technique for evaluation of disinfectant, very important again. Tube dilution and agar plate method is there. Methods याद रखना, डिटेल में पढ़ना है तो पढ़ सकते हो बाद में. Then filter paper and cup plate method is there. Ditch plate method is there. Then phenol water coefficient method is there. Then radial voltage coefficient is there. Then Kelsey Psychis method is there. Then phenol water coefficient test is there. So these are nothing but the test to evaluate the disinfectant. Disinfectant कितना capable है. So to evaluate the disinfectant, techniques for evaluation of disinfectant, एक होता है ट्यूब डाइल्यूशन मेथड और अगर प्लेट मेथड, फिल्टर पेपर और कप प्लेट मेथड, डिच प्लेट मेथड इज देयर, फिनॉल कोएफिशिएंट मेथड इज देयर, केल्से साइके मेथड इज देयर. फिनॉल कोएफिशिएंट टेस्ट इंक्लूड्स, इसके ऊपर बहुत बार क्वेश्चन आ चुका है इसलिए ये लिया है. इन दैट यू आर रेडियल वल्कर टेस्ट, लास्ट ईयर क्वेश्चन आया है इसमें. रेडियल वल्कर ब्रॉथ एंड साल्मोनेला टाइफाई इज यूज्ड एज अ सेंसिटिव ऑर्गेनिज्म. मतलब टेस्ट करना है कि आपका डिसइंफेक्टेंट कितना इफेक्टिव है, whether your disinfectant is effective to kill the bacteria or not. So for that, radial Walker test, which is a test under the phenol coefficient, a phenol coefficient. So RW test, radial Walker test. Radial Walker broth is used and Salmonella typhi is used as a sensitive organism. If radial Walker coefficient of a disinfectant is 1, if RW coefficient of a disinfectant is a 1, if RW coefficient of a disinfectant is a 1, that means it is as effective as phenol. Phenol coefficient, and phenol is taken as a standard, and in comparison with that, other disinfectants they are compared by using phenol coefficient, or that is the test is known as radial Walker test. तो इसमें रेडियल वल्कर ब्रॉथ यूज किया जाता है, साल्मोनेला टाइफी एज अ सेंसिटिव ऑर्गेनिज्म यूज किया जाता है. नाउ इफ RW coefficient of disinfectant under investigation or testing is 1, then it is as effective as. Now if it is less than 1 means it is less effective. If it is more than 1, that means it is more effective than. Next आता है चिक मार्टिन टेस्ट. चिक मार्टिन टेस्ट, ये टेस्ट आप डिटेल में पढ़ो आप, otherwise tomorrow I will take. Then US FDA test is also there. क्योंकि लेना तो ऐसा लगता है कि सब कुछ इम्पोर्टेंट है, पर ये सब नहीं है. इट इज नॉट इजी. लेने के लिए तो सब कुछ इम्पोर्टेंट है, पढ़ना चाहिए. देन US एसोसिएशन ऑफ ऑफिशियल एग्रीकल्चर केमिस्ट टेस्ट, दैट इज AOAC टेस्ट, एसोसिएशन ऑफ ऑफिशियल एग्रीकल्चर केमिस्ट, AOIAC. इसमें रेडियल वल्कर टेस्ट इम्पोर्टेंट है. चिक मार्टिन टेस्ट के ऊपर में भी आ सकता है कुछ. Next, various chemical disinfectant, their properties and pharmaceutical used in the pharmaceuticals. ठीक है, various disinfectant and their properties. ठीक है, पहला है देखो alcohol. So alcohol is used in a 60 to 70% concentration, and a 60 to 70% concentration alcohol is used as a disinfectant. So mechanism क्या है, protein denaturation and damage to the lipid complexes of the. properties क्या है, अल्कोहल क्या होता है, फ्लेमेबल होता है. इट इज अ फ्लेमेबल, वोलेटाइल, एंड इट इज अ स्किन, इट इज अ स्किन टॉक्सिटी. यूजेस क्या है उसके, इट इज यूज्ड एज अ सरफेस एंटीसेप्टिक, डिसइंफेक्टेंट एंड प्रिजर्वेटिव. फिनॉल इज यूज्ड इन द 0.5 to 5% concentration as a disinfectant. मैकेनिज्म है उसका डिस्ट्रक्शन ऑफ सेल वॉल, सेल मेम्ब्रेन एंड प्रोटीन प्रेसिपिटेशन. इट इज नॉट इनएक्टिवेटेड बाय ऑर्गेनिक मैटर. डिसइंफेक्टेंट एंड एंटीसेप्टिक यूजेस. मर्क्यूरी कंपाउंड जो होता है, हेवी मेटल्स, 0.1% में यूज किया जाता है. सो इट कोआगुलेट द cytoplasmic protein of bacteria and it is inactivated by organic matter. So again use as same antiseptic disinfectant. Silver nitrate. Silver nitrate is used in a 0.1% concentration. Protein denaturation or deactivation is a protein deactivation is a mechanism, and it is a weak side, in a microbicide. So weak disinfectant है. It is antiseptic and microbicidal. And next होता है iodine. 2% iodine solution is used. So it causes protein inactivation. It is an oxidizing agent soluble in the alcohol. और ये स्किन वाटर एयर एंड डिसइंफेक्शन है. सो स्किन डिसइंफेक्शन, वाटर डिसइंफेक्शन, एयर डिसइंफेक्शन यूज करते हैं. वाटर के लिए मोस्टली क्लोरीन यूज करते हैं, आयोडीन नहीं यूज करते. क्लोरीन कंपाउंड 5% concentration में यूज करते हैं. सो इट इज अगेन ऑक्सीडेशन of protein and enzyme. It causes the oxidation of protein and enzyme. So inactivated by organic matter and inorganic gases. So mostly used for the disinfection of water, treatment, water treatment, and antiseptic. Disinfectant, antiseptic used for the water treatment. Next होता है articles and respective method of sterilization. ठीक है, ये चार्ट मैंने बनाया है कल. तो कौन से आर्टिकल कौन से मेथड से स्टरलाइज करते हैं, क्वेश्चन ऑफ चॉइस. So say for example disposable syringe. So they are sterilized by gamma radiations and ethylene oxide, and they are used for the powder, fat and oil to inject the powder, fat and oily solution. They are used disposable syringes, they are sterilized by gamma radiations and ethylene oxide. So non-disposable syringes, they are sterilized by autoclave, hot air oven, higher radiations and boiling at a 100, boiling at a below 100. In glass vase they are sterilized by autoclave, water oven. Metal instrument अगर है कोई, जैसे स्कैल्पेल है कुछ है, so that is sterilized by autoclave, water oven and higher radiation. अगर gyroscope है, endoscope है, others anesthetic instruments है, they are sterilized by ethylene oxide and ultraviolet. Surgical dressings है, bowel है, linen है, कपड़े है, and surgical dressing है, linen. That is sterilized by autoclave. Operation theater inoculation wood space जो होता है, that is sterilized by UV radiation. Powder, fats and oil, अगर ये powder, fat and oil, so that is sterilized by hot air oven. Question आ चुका है इसका. Powder, fat and oil, they are sterilized by hot air oven. Then antibiotic solutions and toxins, they are sterilized by sterilization by filtration. Heart and lung machine जो होते हैं, they are sterilized by ethylene oxide. Disposable instrument जो है, they are sterilized by gamma rays and ethylene oxide. Disposable catheter जो होते हैं, gloves होते हैं, and other transfusion set, जैसे police catheter, rails tube, so they are sterilized by gamma rays and ethylene oxide. Sealed bottle ampoules है, aqueous solution, they are sterilized by autoclave. Sealed bottle ampoules जो है, they are sterilized by autoclave. Culture media is also sterilized by autoclave. आपने किया होगा.